Up until this point, we've actually seen how both the respiratory system and the renal system play a role in maintaining blood chemistry. We've seen how the respiratory system and the renal system together regulate the pH of the blood, and we've also seen the renal system's role in maintaining the correct amount of water and sodium and chloride in the blood. Here, we're going to take a look at the role of the endocrine system in maintaining the correct level of glucose in the blood. This is sometimes referred to as blood sugar, but the more correct term is blood glucose. So it turns out that the glucose levels in the blood are maintained within a pretty standard range, and that range is between 90 milligrams per deciliter to about 120 milligrams per deciliter. Those are not numbers that I expect you to memorize if you're in my class, but something around 100 is considered to be within homeostatic range. So let's go ahead and just quickly write that out. So again, your blood glucose levels should be within that range. If your blood glucose levels drop below 90, you can enter a state that is called hypoglycemia. And some of you may recognize that term. If you've entered hypoglycemia, then you don't have enough glucose in your blood to deliver to all of your cells that need it. Remember, the purpose of glucose being in your blood in the first place is so that you can deliver it to your cells, and then your cells are going to break it down for energy, for ATP. So if your glucose levels start to fall and you become hypoglycemic, you might notice a lack of energy, more difficulty thinking, and if they drop dangerously low, like around 30 or 40 milligrams per deciliter, you can enter a coma and it can kill you. So hypoglycemia is something, of course, we want to avoid. Now, the opposite of that is hyperglycemia, when your glucose levels get too high. And it's probably not immediately apparent why hyperglycemia is problematic, but it is, and for a variety of different reasons, actually 2 big reasons. We're going to cover those reasons when we get into our diabetes lecture. So just put a pin in that and know that we will in fact address why hyperglycemia is problematic. So if all is working well, then your blood glucose levels are maintained within this range by 2 different hormones. And those hormones are both secreted by specialized regions of the pancreas called islets of Langerhans. So I'll go ahead and write that down too. And you can see that colored here in the picture on the left. The islets of Langerhans are going to be specialized endocrine cells that secrete hormones. And you can tell them apart because nothing about them looks like duct. So you can see the region that I've circled is an islet of Langerhans, whereas these regions over here, you can see how they have a center to them. Those are all part of small tubes or ducts, and those are going to be the digestive enzymes that are secreted into the small intestine. So that has exocrine function, not endocrine function. So in a previous lecture, I mentioned that the pancreas has both endocrine and exocrine function. Again, this is only an endocrine lecture, so we'll focus on the islets of Langerhans. So the islets of Langerhans secrete actually multiple hormones, and different cells are specialized to secrete different hormones. If you are an alpha cell, then you secrete a hormone called glucagon. If you're a beta cell, you secrete a hormone known as insulin. So let's go ahead and write that down. Both glucagon and insulin are responsible for maintaining our blood glucose levels between 90 and 120 milligrams per deciliter. So what we're going to do now is actually draw out how these 2 hormones have antagonistic effects, meaning they have opposite acting effects on each other, to help make sure that our glucose levels are in fact maintained between 90 and 100 milligrams per deciliter. So if you're going to draw this down with me, which I encourage you to do, you'll probably want a sheet of paper. Portrait style is fine, but you'll need at least half of it to draw all of this out. So we're going to start with our glucose levels in our blood being normal. And I'm just going to use 100 milligrams per deciliter because it's a nice round number. It's easy to wrap your head around. On the right side, we're going to take a look at what happens when glucose levels get too high. On the left side, we'll take a look at what happens when glucose levels get too low. So let's go ahead and start with the too high region. So if glucose levels get too high, let's first think about how that might happen. If your glucose levels are 100 mg per deciliter, how do you elevate them? Well, the most obvious solution is to eat carbohydrates. So I'm going to say that we're going to eat 3 slices of cake. And what's going to happen to that cake when you eat it? Well, of course, that cake is going to go down into your small intestine, where it's going to be chopped up by digestive enzymes and then absorbed into the bloodstream. So about 15 to 30 minutes after eating that cake, your glucose levels are going to start elevating. So this is going to add glucose to our blood. As your glucose levels start to increase, the pancreas is going to sense that and secrete insulin as a response, specifically beta cells. So insulin is now going to be secreted into the bloodstream. When insulin is secreted, it targets a variety of cells around the body, including the liver, the skeletal muscle fibers, and adipose tissue. And it targets those cells to transport more glucose into the cell. If you increase your transport rate of glucose from the extracellular fluid to the intracellular fluid, you're effectively removing glucose from the blood. And when you remove glucose from the blood, your glucose levels in the blood are going to drop. Before writing that out, I'd like to draw that out for you so you get a better visual understanding for what this looks like. So I've drawn part of a body cell here, and I have my plasma membrane, my extracellular fluid, my intracellular fluid. All of these hexagons over here are glucose. This purple thing is an insulin receptor. Insulin is a peptide hormone and therefore uses an extracellular receptor. And then this black square is insulin. Underneath the plasma membrane, I have vesicles. And embedded in the membrane of those vesicles are GLUT4 transporters. This is going to be a type of passive carrier protein. So let's take a look at how this process might work. When insulin levels rise in the blood, of course, they move through the blood and the interstitial fluid. They're going to go ahead and bind to the receptor. That's going to trigger a cascade, and the outcome of that cascade is going to be exocytosis of these vesicles that are stored. And what that's going to do is that actually inserts the GLUT4 transporters into the plasma membrane. So we've now inserted new glucose transporters into our plasma membrane of our body cell. And again, this could be a liver cell, this could be an adipose cell, this could be a skeletal muscle cell. So by adding more GLUT transporters, what we're going to do is we're going to be able to transport more glucose from the extracellular fluid into the intracellular fluid. And by doing so, notice that we are now reducing the levels of glucose in the blood. So the goal of insulin is to lower the glucose levels in the blood by simply moving that glucose into the cells. So now that we've seen this in action, let's go ahead and add it to our negative feedback loop. I'm going to add something to this side that happens probably in all the cells, but especially happens in the liver. So one of the major targets of insulin is in fact the liver. And what the liver does is not only transports glucose, but then takes that glucose and synthesizes a complex carbohydrate from it called glycogen. And I'll show you what I mean here. If we come back to this image and assume that this body cell is in fact a hepatocyte or liver cell, then what insulin also does is synthesizes a complex carbohydrate called glycogen. Now you might notice that in synthesizing glycogen, the levels of actual glucose in the cell decrease, which guess what? Is kind of a sneaky way to create a more favorable gradient to draw even more glucose in, which then you'll just keep attaching to that glycogen, growing glycogen chains. So while this seems to happen in all of the cells that are targeted by insulin, it's especially prolific in the liver. So let's go ahead and add that to our negative feedback loop. All right, that concludes our look at what happens when the glucose levels get too high. A series of steps that prevents hyperglycemia. Now let's take a look at the opposite. What happens when the glucose levels get too low? Now, how could the glucose levels get too low? Easy. So if you're not eating and you're using up all of your glucose, then your glucose levels in your bloodstream are going to get low. But let's go ahead and kind of take this idea to the extreme by saying that we're running or doing some sort of exercise and fasting at the same time. So now that glucose levels have dropped, the alpha cells are going to sense this and secrete glucagon. So remember, the alpha cells are also located in the pancreas. That glucagon's major target is going to be the liver. So remember that the liver previously synthesized glycogen from glucose in your last meal. Well, that glycogen represents a storage form of glucose. So glucagon is going to tap into that liver's supply and ask the liver to liquidate that glycogen, make more glucose, and dump it out into the bloodstream so that our blood glucose levels come back up. So now we've created glucose by cutting up glycogen, and so that glucose is added into the bloodstream to try to bring our glucose levels back up. So together, these two hormones help maintain our glucose levels around 100 milligrams per deciliter. So let's address what might happen if the glycogen levels in your liver become depleted, which happens usually about 6 to 8 hours after fasting. So your liver doesn't have an infinite supply of glycogen, and so you'll burn through your glycogen in 6 to 8 hours, and then what? Well, the next place that the body is going to go is going to be proteins. And it's actually going to cut up proteins and chop them up into amino acids, and from there re-engineer those amino acids as glucose. So it will build glucose from amino acids in a process known as gluconeogenesis. So let's go ahead and put that in. So the phrase gluconeogenesis tells you exactly what it's doing. We have gluco for glucose. Neo means new, and genesis means to create. So we are creating new glucose from amino acids. So if you have a variety of amino acids in a protein, you can cut up that protein into the individual amino acids, and then you can get rid of the nitrogen, and through a variety of metabolic steps create glucose instead. And you'll produce ammonia and urea as a byproduct of that. So if somebody is starving for glucose, they can actually break down so many proteins that they smell like ammonia. It's true. Gross, but true. Okay, so typically your hunger drive is going to, try to make sure that you don't go into this route, but if you are not eating and your glycogen reserves get too low, then your body will start breaking down its own proteins, its own muscle, its own bone, in order to make new glucose. In addition, it will liquidate fat. So it will add fatty acids to the bloodstream. Now, not all cells like to use fatty acids as their preferred source of fuel. So for instance, the brain, for whatever reason, does not like using fat as fuel. Muscles, however, are okay with it under certain circumstances. So fatty acids can be used to try to help bridge the gap while you're waiting for your next meal, but cannot be used as an exclusive fuel source for the body because certain cells simply just don't like using it. I'll see you for the next lecture on diabetes.